brains are mysterious. They're complicated systems that do mysterious things, and we don't really understand how they work. It's interesting. We each have one. We each have a brain, and yet somehow, though we use them every day, it's unclear how they're doing the magic that they do. The same might be said of neural networks. Neural networks are mysterious. They're complicated systems, and we don't really know how they work. And you've probably trained one in your time, and it's not clear how it's doing what it's doing. So my research really combines those two things and asks, if we have these two mysterious systems, can we use one to help us understand the other? And can we use one to help improve the other? So when I talk about a neural network, what we're interested inside of the neural network is the representations that it learns. So as a byproduct of the task we train a neural network to do, for example, like object classification, it takes some input and produces a prediction about it. And as a byproduct of that predictive process, it learns representations. Those representations facilitate its predictive process, and it's just a vector of real numbers, so just a list of numbers. But that list of numbers is constructed so that it can do the predictive task it's trained for. And the representations exist at these hidden layers of the neural network. Inside the brain, there are also representations, and they are made by groups of neurons working together. So as your groups of neurons work together, they encode the things that you perceive, they encode the thoughts that you have, and they work together to support the sorts of things that you want to do with your brain. So if you want to do object classification, you can do that, but you can also do lots of other things like give a talk. And we're going to record brain activity with brain imaging. And once we get a brain image, it's just a vector of real numbers. So it's a representation in the brain, but it's still just like a vector of numbers, just like a representation in a neural network. So in a lot of ways, there's comparisons between the two, and we're going to find the connections between these two representational states. So we have neural networks, and we have brains. They're both doing something, some algorithm, and as a byproduct of the algorithm, they create representations and they use representations. So we'd like to peer into each of them to help us understand what they're doing. Two systems, complicated, but we can see these artifacts of the algorithm. As they're moving through the steps that they need to compute to do the task, they're creating these sort of waypoints along the way, and we can measure those waypoints in both cases, and we're going to use them to try to figure out what is the algorithm that these two systems are doing and what are the parallels between the algorithms. So you may have heard of Marr's levels of analysis. Marr was a famous psychologist, and he thought of how to break problems down into pieces so that we could understand them in their entirety, so that we could really fully understand the system we were trying to study. So he characterized problems at 3 different levels: computational, algorithmic, and implementational. And we'll go through each of the levels in turn. Today we're going to talk about an example task. Let's talk about object classification. So when you look at a picture, you can tell what's in it, and we can train neural networks to do that too. So here we have a column for brains and a column for neural networks. And so I put an asterisk beside object detection, object classification for brains, because clearly your brain's doing a lot more than just object classification. You're doing other things too. But for now, let's just think about the part of your brain that does object classification. So on the computational level, we can train a neural network to do the same task. And so we have a parallel between the brain and the neural network at the computational level. And then at the bottom, we have the implementational level, and we have neurons and transistors. So brains work using neurons, computers work using transistors. So we don't have a parallel there, but there is some level at which we can consider those to be transferable or interchangeable. So what we're really interested in asking questions about is this algorithmic level. Can we figure out the algorithm the brain is using to do object classification? And there's been a lot of research, and they've made a lot of progress. We understand a lot of what the brain is doing during object classification, but we don't understand everything. The interesting thing about a neural network is actually we can write down exactly what it's doing, right? We know the exact function the neural network learns during its training process, right? We know all of the parameters. But at the same time, we actually don't really know what it's doing, right? It's hard for us to describe what the intermediate functions are doing inside of a neural network. So I put a question mark for the brain's algorithm. I also put a kind of a question mark for the neural network. So because we know what it's doing, but we also don't really understand it. So what we would like to do is to detect the artifacts of the algorithm. So this I actually attribute to my colleague, Mike Dawson. So having a joint position between computer science and psychology has actually been really cool. And one of the things that it's exposed me to is these new ideas. The artifacts of the algorithm are the things that you can measure that the algorithm is creating as a byproduct of what it's actually doing. And so we would like to measure the artifacts of these two algorithms, the one in the brain and the one in the neural network, and ask if there's a relationship between those artifacts. So in the brain, we can measure the neural activation using things like brain imaging. And in a neural network, we can measure the hidden representations, which are the intermediate layers of the neural network. And now we're going to ask, do those artifacts align? Is there a relationship between the artifacts of the algorithm in the two systems? And if there is a relationship between those artifacts of the algorithm, then we know they're doing something similar, right? So now we're going to take a little break, and we're going to talk about neuroimaging. We have to talk about neuroimaging in order to talk about how we can actually record the artifacts of the algorithm inside of your brain. So this is fMRI. You've probably heard, if you've heard of any brain imaging, you've probably heard of fMRI. Often makes the news. So fMRI is basically a large magnetic donut. We put your brain inside of your head, and we bounce magnetic fields off of it, and it records the changes in blood oxygenation in your brain in a response to your neurons' activity. So the more your neurons work, the more oxygen that they use, and we can detect that with BOLD, with an fMRI. It has good spatial resolution. You can get down to a millimeter cubed. Now, in a millimeter cube, there are still tens of thousands of neurons. So it's like we are not recording single neurons here. We're still recording groups. But it's pretty good spatial resolution, about a millimeter cubed. It does not have great time resolution, which means we get one sample every 2 seconds. So if you want to study something that's a fast-moving process like language, fMRI does definitely have some drawbacks. What comes out of the fMRI is basically a 3D picture, and instead of pixels, we call them voxels because it's a volumetric pixel. So each one of the voxels represents one of those 1 millimeter cubed areas of your brain, and we look at them in these slices. These slices on the bottom row is actually the top of your brain, and as we're moving up in the picture, we're actually moving down through your head. So this is what fMRI looks like. The bright colors would mean that the bold signal is higher in that particular location, so we can see which parts of your brain are working more or less in response to a particular stimulus. This is EEG, or electroencephalography, another popular method for recording brain activity. So here we have these little sensors we put on the outside of your head, and they record the electrical activity of the neurons that are firing inside of your brain. But we can actually measure that outside of your head, which I think is remarkable and amazing. The time resolution is great. We can get 1,000 samples a second, so it's great for studying things like language. But it doesn't have great spatial resolution because we're measuring through your skull and your scalp, and so there's some interference there. But EEG systems are much cheaper, on the order of tens of thousands of dollars, whereas an MRI machine is millions of dollars. So somebody like me can collect EEG with my own machine in my own lab. A nice thing about EEG is it can be collected anywhere. So we could collect EEG in this room, although it's a little bit better if you do it in a quiet location. So what comes out of the EEG is that little graph there. It's just a bunch of sensor readings and tells us which parts of your brain are creating more or less electrical activity at a particular point in time. And the third kind of brain imaging we're going to talk about today are direct neural recordings. So these are invasive and they're implanted, which means you actually have to open up a person or an animal's skull in order to put in this recording. So that's a Utah array there, and it's just like a little square with a bunch of needles poking out of it. They just place it on top of the brain, and then they can record from the neurons that are directly underneath that one little patch. So it's great time resolution, great spatial resolution for the very small little piece of brain that it covers. So for the most part, these are used in animal experiments. So they would use animal models in order to do direct neural recordings. So here's a picture of a monkey brain. And that little yellow rectangle is the Utah array, the array that we talked about. And it's going to record the neurons that are directly under that patch. And what you see on the right-hand side is what's called a raster plot. And it tells us essentially how many neuron spikes do we see for a particular neuron and a particular point in time. Each one of those black dots says that this neuron fired at this particular time. So it tells us for a particular image, if the monkey's looking at an image, how many times did this particular neuron fire in response to that image. So each one of these things is a way to record brain activity, and each one of these things is just a vector of numbers. And so we're just going to turn them into vectors of numbers. We're going to flatten all of those sensor recordings. It comes in as a matrix, just turn it into a vector. The fMRI comes in as a matrix, we just turn it into a vector. Everything's a vector, and it represents something about what the brain is doing under the hood. So now we're going to ask, using these ways of studying the brain, can we find an alignment between the artifacts of the algorithm for vision? So we'll start with vision, and then later on in the talk, we're going to talk a little bit about language. So you're probably familiar with ImageNet. It's an amazing data set that was created about a decade ago now. And it has millions of images covering 1,000 different classes. The diversity of images in this data set is actually really remarkable because they're just pulled from the internet. So these are just pictures off of Flickr. The picture people just took out in the real world. So they're not composed in any special way. They're amateur. They're not, the thing of interest might not always be right in the middle of the picture. And so it's a really diverse data set, and it's actually really hard, it was really hard to train accurate models on this particular data set, but neural networks really changed that. And made it able for us to get extremely high accuracy on this data set and differentiate between thousands of different types of objects. And the way that we did that, I had nothing to do with it. The way that other people did that was with convolutional neural networks. So convolutional neural networks take as input an image. And then they perform a bunch of computations on that image and produce as output a prediction of what's in the image. In this particular example, these blue squares are the intermediate representations, and they represent what the computer algorithm is computing on its way to making that prediction. They support this predictive process. They are the representations that we're going to talk about. So let's look, so here this says, what do the neurons represent? Here I mean the actual neurons inside of the neural network. So let's choose a particular neuron in this neural network. Let's say this particular neuron exists at this layer, and we're going to call it H. We would like to know, how can we make H's value as high as possible? We're doing this because we would like to know what H represents. So on the pathway from input to prediction, H is doing a job. We'd like to figure out what that job is, and one way that we might be able to do that is by looking at, when does H get really excited? For what kinds of images might we see really big values for H? So in order to do that, we'll actually take the input image and start with something that's all gray, and then we're going to take a particular pixel and either turn it up or down depending on how H reacts to that change. So we'll turn this pixel up and this pixel down, so on and so forth for a bunch of pixels in the image, altering the image so that we can make H fire as maximally as possible. So we want to know, what is the input stimuli that makes H fire maximally? And when we do that, for the first level of a convolutional neural network, the things that make neurons in that first layer fire maximally are things that look like this. They look a lot like edge detectors. They're looking for places in the image where something goes from being very light to very dark. So that's what these particular neurons are tuned for. They're looking for those patches of the images where we see big changes, and those correspond to edges in the visual field. And as we move through the neural network, here the bottom is CNN1, and as we move up, we're going farther into the neural network and getting into more complicated layers. And each one of them builds on the layer that came before. So CNN2 has the features from CNN1 as the input. And you see that it's using it to make more complicated things. It's detecting more complicated features. So the neurons in CNN2 fire maximally when they see things like curves or spirals. And then CNN3, we're getting even more complicated things, the spirals, the squares, sets of dots together. So you can see how each one of the layers is building on the one before it to create a more complex picture of what's actually going on in the image. And now we're moving up still through the neural network. This is layer 6, 7, 8. This is AlexNet, if you're familiar with it. Layer 8 is actually the prediction layer. So layer 8 is actually the layer where we would say, what is the object in this image? And so one of the neurons actually corresponds to the concept that's labeled here. So for the concept mushroom, you can see that the things that make this neuron fire most maximally actually do look a lot like mushrooms, right? Cups, gazelles, vultures. So you can see that the things that are making these neurons fire maximally look like the thing that should be. And so as a byproduct of that, we can look at the layers before that and sort of infer what kinds of things they're looking for. So I see some dog faces on the middle row on the right-hand side. You might see something different. So that tells you a little bit about what the neurons are looking for in the brain, in the neural network. And the neural networks were constructed to mirror what we knew about the brain. So we knew there was a hierarchy of processing in the visual system. V1 is one of the earliest layers of your visual system that it receives input from your eyes. And then V2 builds upon what's computed in V1. V4 builds upon what's computed in V2. IT builds upon what's computed in V4. So it is the same hierarchy of processing, just like we see in the neural network. And so neural networks were really inspired by what we knew about the processing, the hierarchy in the visual system. So now we might ask, do the artifacts align? Is there a relationship between what we can measure in the visual system of the brain in that hierarchy and the sorts of things we see in the representational features of the neural network? So we have the same hierarchical structure. Is there a relationship between one or the other? So for a particular ROI, we'll measure the activation in that particular location. And then we want to know, for this particular spot, how similar is it to these other layers? So we'll compare each one of these ROIs to a particular layer. And what they've actually found is that the hierarchy and the similarity are sort of mirrors of each other. And I'll walk you through that result now. So what they did is they took images and they showed them to people in an MRI scanner and collected brain imaging data while they did it. Here's fMRI. They showed the same images to a convolutional neural network and measured the hidden representations of that neural network during the computation. And this neural network is trained to do object classification. So we have 8 CNN layers, and there are 7 vision-related areas in the brain. Each one of these is just vectors, so we have 8 vectors per image from the CNN and 8 vectors per image from the brain. 7 vectors per image from the brain. So this, again, fMRI data. So are we recording from single neurons? No, we're not recording from single neurons. We're recording from groups of neurons, but we're still looking for that relationship between the representational spaces. So there are 8 CNN layers and 7 vision-related areas, and what we're going to do is train 56, here they're just using regression models, to predict the CNN representation as a function of the fMRI data. So the input to the regression model is that vector of fMRI activations, and the output is a prediction of the hidden representation for a particular layer of the neural network. And so what we'd like to know is, which predictions are most correlated with the true CNN representations, or which one of the regression models is the best? So this is a kind of a busy graph. On the y-axis, we have correlation, and on the x-axis are different areas of the brain. And there are 8 graphs here. Each one corresponds to a different layer of the neural network, CNN1 being one of the earliest layers of the neural network, and CNN8 being the output layer, the very final layer of the neural network. So as we move from bottom to top, we're going in increasing complexity, and left to right is increasing complexity in the brain. And so what you can see is that for some of the least complex CNN layers, like CNN 2, 3, 4, we have higher correlation to the least complex areas of the brain. And the reverse is also true. As we get into the more complex layers of the neural network, 7 and 8, we start to see higher correlation to the higher order visual areas of the brain. So there is this relationship between this hierarchy in both systems. They're computing something, and the artifacts of those algorithms are aligned in a way that respects the hierarchy. So early layers of the visual system and early layers of the CNN are aligned, and the same is true for later layers. So there is a relationship between the higher order vision areas in your brain and also the higher order areas of the CNN. So that's interesting, but is it useful? So one other thing that had come out of research previous was that not only was there this relationship between the layers, also as the CNN became more accurate, the relationship came stronger. So a more accurate CNN would have better correlations to the brain on average. So we actually were interested in asking the reverse question. What happens if we make the representations more correlated? Do we end up with a more accurate CNN? So this is work with my collaborator, Joel Zilberberg, who's actually here today, and his PhD student, Callie Federer. Our postdoc, Richard Jerem and Cassidy Pirlo, both continue this work today. So we use CornetZ, which is a great neural network for this sort of thing because it actually has direct mappings onto the brain. So it has a V1 area of the neural network, V2, V4, IT. And what we're going to do is we're going to record the activation, the representation from a particular layer of the neural network. Here we're going to choose V1. We're going to, for each one of the images that we show the neural network, we're going to write down, we're going to record that particular representation from that particular layer. We're going to do the same thing with neural data. We're going to show these images to a monkey, and this is pre-existing public data. They recorded in this monkey from the V1 area, and so again, we just have a vector that represents the brain's representation of that particular image. Now we're interested, again, artifacts of the algorithm, we're interested if these artifacts align. We know previously that they do, but here we're actually interested in saying, if we get the neural network to be more like this representation, does it become a better neural network? Does it become more accurate at its category label prediction? So what we get here are representational similarity matrices. So these are matrices that just compute the pairwise similarity between the representations in each one of the spaces. So on this side, here the orange one is the representations for a neural network. So for a particular image, say this picture of a cat, we're going to compute its similarity to all of the other images. So we'll say how similar in the neural network's representation is this cat to, for example, a cow or to a train. And say the neural network thinks one is more similar than the other, its similarities will show up in this representational similarity matrix. We're going to do the same thing for the neural data. We're going to say for this picture of a cat, is the neural recording, is the brain's activation in response to this picture of a cat more similar to the cow or more similar to a train? And what we would like is for these two matrices to be similar. We would like both of the representational spaces to have similar structure. If the brain thinks that two things are similar, we would like the neural network to also think that those two things are similar in representational space. So we're going to train a neural network to minimize categorization errors. That's the typical way that we train a convolutional neural network. But we're also going to introduce a brain constraint where we're going to encourage the neural network to create representations that respect the similarity patterns that we saw in the monkey data. So we want these matrices to be as similar as possible, and we can actually do backpropagation through the neural network to make these representations fit to be more close to what we saw in the brain. Here's the obligatory math. So it's just the square difference between what we observe in the monkey and what we see in the neural network, minus the cross-entropy loss. So what we saw was small but robust improvements in accuracy when we introduced this new constraint. So we saw that the network got even more accurate when it created representations that looked like what we saw in the brain. So here r equals 0 is the baseline. It means that there's no impact from the neural data, the monkey recordings at all. And as the values of r get larger, we see an increase in performance and then a gradual decrease in performance as r gets too big. So for a non-zero value of r, we see better convolutional neural network performance. Another interesting thing is that we also saw better superclass accuracy. So every neural network makes some mistakes. And the question is, are the mistakes that the neural network makes are they reasonable? Are they sort of the sort of thing that would make you go, so if we confuse a cat and a cow, maybe that's not so bad because cats and cows, at least they're both animals. But if we confuse a cat and a train, something has gone very wrong in our neural network. Something weird is happening. So we can also measure superclass accuracy. So here on the y-axis is the percentage of the errors that we did make, what percentage were at least within the correct superclass. So higher is better. And we see for a non-zero value of r, which is on the x-axis, we see higher accuracy, superclass accuracy, meaning that when it makes mistakes, at least they're less embarrassing mistakes. So we saw that artifacts of the algorithm align for vision, and also when we make them align more strongly, the CNN, the convolutional neural network, gets even better. We'd also, in my lab, we also study language. And so we've done a similar sort of thing for language. We use brain activity while people read or listen to language. And we compare them to the hidden representations of neural models of language, neural network models of language. So here is our picture that we were doing before. Remember, we had the convolutional neural network on the top, and we had brain imaging data and the 7 vision-related areas. So we're going to use the same structure, except now, of course, we can't use a CNN because we're going to do language, and we're not going to use the vision-related areas of the brain because we're doing language. And we're going to show the word cat, and so it doesn't make sense to use the CNN or the vision-related areas, so we're going to wipe them out. We're going to take the word cat, and we're going to show it to a neural network. Here, this neural network is skipgram. It's a shallow neural network, and it's trained to essentially predict word co-occurrence. And as a byproduct of that predictive process, it creates hidden representations just like a convolutional neural network. And those hidden representations can be assigned to particular words. So let's say that this purple vector represents the hidden representation for the word cat. We can also show the word cat to a person or have them listen to it and then record the activity in their brain in response to the particular word. And then just like we saw before for the vision experiment, we're going to train regression models that can predict the skipgram vector as a function of the brain images. So the input is the brain image, just like we talked about. The output now is some prediction of a language representation. So on the previous slide, I showed you skipgram, but you can actually use lots of different representations from different language models. You've probably heard of transformers, RNNs, LSTMs. All of those actually have been used in this process to compare to people reading single words or sentences. And we found that it works really well for adults doing all of these tasks. If you have a person come and sit in a scanner and you record their brain activity, we get the same relationship of artifacts of the algorithm align in the same way as they do in vision in language. And so that's an interesting result, but we were interested in changing this question slightly. So instead of asking, does this work well for adults, we asked, does this work well for babies? Is it true that a baby, before they can even talk, has a representation of language that's similar enough to an adult's that we can use this same sort of decoding paradigm? So you call this decoding when we take as input a brain image and try to predict what it was the person was experiencing. So this is in collaboration with Jennifer Campbell and Janet Worker at UBC. They collected this brain imaging data, so we were absolutely, in awe of what they did. They had the babies into their lab, dealt with all of the things that comes along with babies, keeping them happy long enough to do the experiment. So we are, forever in debt to those two women. Rohan is my master's, now PhD student, and he's probably here somewhere, who worked on this project with me. And Janet actually was one of the pioneers of studying language in babies. And originally, she used a looking paradigm to do these studies. So back before brain imaging was as cheap as it is now, couldn't just hook babies up to EEGs or MRIs. We had to do something a little smarter. They did a looking time paradigm, which means that they would say something like, where's the cup to the baby? And then they would show them two pictures. Here's a picture of a dog and a picture of a cup. If the baby understands the word cup, they will look at the cup longer than the dog. If they don't understand the word cup, they'll look equally at both. So we can tell what words does a baby understand using these kind of looking time paradigms. And it's remarkable what they've found. Using these sorts of simple paradigms, you don't need brain imaging at all. So we can be a little bit lazier because now we have brain imaging. So here's our experiment. So we have a parent come into the lab with their baby. The baby sits on their lap, and we collect EEG from, this should say 9 and 12-month-olds, 9 and 12-month-old babies, and we get an EEG recording. And the experimenter in this case was Jen. She's sitting in the room with the parent and the baby, and she's saying a set of words. So she'll say like the word banana, and then on the screen will appear a picture. Sometimes it's a banana and sometimes it's not. And we're going to record the EEG and particularly pay attention to the EEG that's collected after the time Jen says the word banana. So here's what it looks like. Jen says banana. We see a picture of a banana. Jen says the word dog. Uh-oh, we saw a picture of a spoon. So we set it up this way because there's a brain response called the N400, which happens in the brain in response to stimuli that don't match what you were expecting. Sorry, but for today's experiment, we're actually going to just use the brain imaging data collected just after Jen said the word of interest. So we're going to completely ignore this other part of the data. So they did see images. Today, we're not talking about that. We're just talking about the brain imaging data collected after they heard the word. So they're going to hear the word cat. We're going to get EEG data while they heard the word cat, and we're going to represent that as a vector of numbers. We're going to also pass the word cat to our neural network and get out the hidden representation for the word cat. And then we're going to train some regression models in order to predict that skipgram vector, that representation for the word cat from the EEG data. I'm not going to talk about it today, but the way we measure the accuracy of these systems is 2 versus 2 accuracy. You could just think of it as like, how good are we at predicting that hidden representation? And chance is 50%. Yes. So here is the graph that's going to show the results that I'm talking about. On the x-axis is time. 0 is the onset of Jen talking. It's a little fuzzy because the onset of speech is not always perfect, but it's sort of around that 0 point. And on the y-axis is 2 versus 2 accuracy. Remember, chance is 50%. So what I'm going to show you is the decoding results, the accuracy, using brain imaging data from 9-month-olds and 12-month-olds. So here you can see, so along the bottom, there's the green and purple stripes. those stripes represent points in time where the line is above chance. So we can, with above chance accuracy, tell the difference between 9 and 12-month-old. So we can tell what word a 9-month-old or a 12-month-old baby is hearing using just their EEG data. So this is additional evidence that babies actually understand the words that we're saying to them, and also that they have representations of those words that fit what adults would sort of understand about the world. So it's not just that they understand cup to be a particular graphic image on a screen. They actually understand cup is more similar to, say, bottle than it is to cat. In addition, we saw that there was a significant difference between the 9-month-old and the 12-month-olds. It's a little hard to see. I think maybe you can see it a little bit on the screen. The later portion of the graph is highlighted pink, and that's where there's a significant difference between the two lines, meaning that here it's easier for us later in time to tell what word a 9-month-old is hearing, versus what a 12-month-old is hearing. And people are often surprised when I show them this graph. They think that 12-month-old babies should have stronger and longer representations of words. But actually your brain imaging data would fall off about like this 12-month-old's does. When you hear a word, a representation pops into your head and then it's gone. Because you're good at this. You know how to pull up words in your mind. 9-month-olds takes a little bit more time. They're figuring it out. This is my hypothesis. They're figuring it out. It takes them a little bit longer to get it into their head, and they have to hold it for a little bit longer to sort of finish their processing process. So we see a significant difference later in time, but for both groups of babies, we can tell what word they're hearing using the EEG data. Now, so I showed you there's a significant difference between these two groups of infants. And so one question that you might want to know, is the representation similar? So the way that 12-month-old babies understand the word cup, is that similar to the way that 9-month-old babies understand the word cup? In order to answer that question, what we can do is train a model using one group of babies' data and then test it on the other group of babies. So that tells us if we used 9-month-old babies to find a pattern that matched cup, if we tested on 12-month-old babies to see if it still matches, we would know that their representations are similar. So this is the same kind of graph. We have 2 versus 2 accuracy and time on the x-axis. And here is the results. 9 months, training on 9 and testing on 12 is green, training on 12 and testing on 9 is purple. But the main thing to notice here is that along the x-axis, there's those little dots, and that tells us that with above chance accuracy, we can tell what word was being heard by one group of infants, even though we trained on a different one. So that means that the representations are shared. They're similar enough across these 2 groups of babies that we can train and test across the groups. So even though there's actually a lot of language development that happens between 9 and 12-month-olds, we actually see similarities between the representations in those 2 groups. Something that people often ask me when I show them this work is, but you use Google News. So the skip-gram model that I talked about is trained on Google News, which is lots and lots of news article transcriptions, and they trained the word vector model to do the word co-occurrence prediction. Babies don't usually listen to news programs, certainly not as much as adults do. How could we possibly have used news articles in order to train this model? Does that even make sense? On one level it does not, but on another level it kind of does. So here's a dendrogram of the word vectors that come out of this model trained on Google News. A dendrogram just tells you which vectors are the most similar. So on the x-axis are all the words, and you can see that words that are more similar are grouped together in a way that you would probably think a baby would also agree with, if a baby could agree with this sort of thing. Dogs and cats are next to each other, cookies and crackers are next to each other, juice and milk are next to each other, moms and babies, which are the only 2 humans in the data set, are next to each other. So even though this data was trained on Google News, it understands the world in a way, for this simple subset of words, that's probably pretty consistent with what a baby would know. So it's trained on data that absolutely a baby has not been exposed to in their lifetime, but it creates word vectors that's, I think, reasonable for the task that we're actually trying to do. So we can detect word meaning in infants, which I was blown away, totally surprised. This was the coolest thing. I was so excited to work with Janet. If you want more details on that, we have a poster during session B. And our future work, one of the things we're interested in doing is expanding this to try to understand how children's representations of language change. From infant to children and then from children to adult. How do the way that we understand the world affect the way that we understand language? And can we see those representations in brain imaging data, or what we're actually doing here is collecting behavioral data? So there's more details about that at our poster, also session B. I'm also really interested in trying to expand this to story listening. So if you've ever read a story to a baby, you know that they are just glued to the book. And I think it would be really cool to try to see how their response to sentences and story structure matches that of adults. So sort of the similar sorts of experiments have been done with adults for different stories. I'd like to know if the same sort of findings hold up in infants. So I showed you that the artifacts of the algorithm align for brains understanding language and neural networks understanding language for some definition of understanding language. I showed you that there was an alignment in the artifacts of the algorithm. So there is some way in which our models represent language that's aligned to what brains do when they understand language. And I think there's value in understanding how that works in adults, but there's also value in trying to understand how that happens in infants who are in the process of acquiring their very first language. They're in the process of starting to figure out what words are and what words mean and how they fit together. And so understanding that process of learning language, I think can help us build neural network models of language that learn better. That's my hope anyway. And I think I would like to encourage you to think about your favorite task. So what is the task that you're working on? Is there a way that we could measure it in humans, a similar task in humans, and then compare the artifacts of the algorithm? Would there be an alignment? And if there was, would that help you in some way? So I think this is a sort of an interesting way to think about two different ways of studying a problem and thinking about how understanding one system might help you to understand the other. That's all I have today. Thanks very much. I'll take questions.
Thank you, Alona. Very interesting work. So we have time for questions right now. And the way the questions are going to work is there's 4 microphones here, so people can come up to the microphones and ask questions. But I just want to emphasize, come with questions. So we want to see questions, not comments or commentary. But brief questions will be great. We have about 15 minutes, so I'll let Alona run it from here.
Great, thanks.
Over here.
Hi.
Hi.
So my question is, like, when we feed an input to a neural network, whether it's an image or a text, it's a very clean input. But for the brain, even if you're looking at a picture or reading a text, we are always constantly aware of our surroundings, and there's a lot more computation going on in our brain. So in this study, do you try to filter out that, like do some calibration, baseline, and try to filter out other computation?
Okay, so there's lots of interesting questions wrapped up in that question. One thing is like the brain's doing lots of stuff even when you're reading. That's absolutely true. And so part of what we have to do for, especially for things like MRI, is take into account some of those other processes. So, subtract off some sort of baseline that tells us like this is the part of your brain that's working even though you're just breathing. So there's definitely parts of your brain that are doing that have activity when you're reading that have nothing to do with reading, right? That have to do with like keeping you alive. And so we would like to sort of subtract those off, but it's also true that we're looking for the parts that are correlated with the stimuli. And so in some ways those would sort of fall away because, the brain activity related to breathing should be fairly constant over the whole experiment. And we're looking for the things that are in particular aligned to the stimulus that we're interested in. Another thing I think you're sort of getting at is that there is a lot going on when we do something like reading. And when I show you these really simple experiments where it's just one word and like, what does that even mean? It's true that reading is a much more complex thing when it's lots of words all in a row that go together. And I think there's a really big push towards working on language in more complex settings. For example, there's fMRI studies that used Harry Potter and had people read an entire chapter of Harry Potter or listen to Alice in Wonderland and look at brain responses to more complex text like that, because there's a lot more going on when you're reading text that's just not a single word. Does that cover your question?
Yeah, it does. Can I ask one more question?
Sure.
So I read a paper from Tom Michel a few years ago in which he has compared different language models, and he found that representation learned by BERT model is better than other models. So did you also find something similar with the image? Like some model learns representation which is more similar to the brain or data or readings that we get from the brain?
Yeah, so this goes back to what I was saying, that like CNN models that are more accurate are more correlated to brain imaging data. And language models, it's also true for some language models, as they become more accurate, their representations become more correlated to brain imaging data. Yeah. Oh, hi.
So I'm interested in the difference between the brain and models like neural network to understand high-level concepts on Seaborn concepts. So for example, if you consider systems like DALL-E 2, they can draw amazing images based on representation. So if I tell them to draw a frog swimming in a coffee cup in a surrealistic way, they would produce great results. But if I tell DALL-E to draw 17 apples, then it wouldn't know what to do because it doesn't understand the concept of numbers. But if I'll tell you to draw 17 numbers or even a million numbers, then you'll know what to do. It will be hard to draw a million apples, but neural networks can't currently face these issues. So I wonder if the problem is with the representation differences or with the algorithm, or what do you think about that?
So I want to push back on one thing that you're talking about, which is that sort of neural networks don't understand numbers. Humans actually are very bad at estimating numbers beyond 5. So in some ways, you don't really understand numbers either if you just see an image quickly. So we have to give them a little bit of credit. I actually think some of what's showing up there is actually a good mirror to what people do. Not great at understanding groups of things. We can't enumerate large groups of items. But I think what you're getting at might be something more interesting. Like, what could we make an experiment where we ask somebody to picture something, and then they've sort of done this in some ways, use the brain imaging data to estimate how they're sort of creating that imagery in their head, and then can that be used to make DALL-E better? That would be an interesting experiment. There's actually been some work showing that even during imagery, the representations that show up in the brain in response to being told, like, just think of a leopard, those responses in your brain are actually correlated to what you would create in your brain if you saw a picture of the leopard.
Yeah, thank you. I think that if I am trying to draw 17 apples, then even if I can't imagine that in a good way, I'll still be able to accomplish this task.
Yes, I agree. And you would probably, at least you would draw many, many apples, even if it wasn't 17.
Yeah. Thank you very much.
Sure.
Hi, Ross Mitchell. Fantastic talk. Just super, super cool. So I have two questions. One is, have you looked at how the latent space representation changes, for example, between a 9-month-old and a 12-month-old as they learn? Like, is there structure in the latent space? You know, the famous paper, king minus man equals queen minus woman. Is there that kind of structure in the latent space, and does it change? And the second question is, have you looked at feeding some of your latent space representations into the traditional neural network to help it learn as you train, sort of cross-training across domains? You know, we can, if you want to, you can train a neural net to look at an image and look at words, and it will improve its performance. I wonder if your neural nets with your latent space representation that you've learned would help improve training that way. Thanks.
Yeah, there's some really cool networks that have been trained in parallel with captions of images and the images themselves, and I've done really cool things with that. So there's some of that. We are also super interested in trying to use language representations from the brain to train better language models. So that's definitely on the horizon, but I'd say that's early days. The visual system is much better understood than the language system in the brain. A good first question too. Does anybody remember the first question?
Yeah, I do. Does the latent space representation change between 9-month-old and 12-month-old?
Oh yeah, so that's what we were after, right? We were like, so there's actually big reorganizations that happened during that time. This is based on what Janet had told us. So we actually expected that the experiment of training and testing between the groups would not work, but it did work. And one of the things we were interested in doing in the longer term is looking at how the representational spaces sort of morph over time. So we're collecting brain imaging data. We collected it in infants and then coronavirus, thank you very much. But the idea was to also collect it in, say, school-age children and also in adults and compare across them to see when are those big shifts. Like, can we actually see those big shifts when we do the brain imaging data collection itself? Yeah, so you're exactly right. That's where we were going. Maybe I'll ask your question.
Sure. Yeah, thank you so much for the really fascinating and enjoyable talk. That was great. Maybe this is related to the response you just gave to the previous question, but I was really surprised that the decoding works across age groups. And there were some periods when the 12-month to 9-month was better decoding accuracy. So what does that say about the representation in 9 months? Mm-hmm. How do you interpret that result?
Yeah, I would try to flip back, but I don't know how many slides that is. Yeah, so I have often, it's kept me up at night. Why would it be better? So in some of the cases, we can get above chance accuracy by training in one time window and testing in another when one of those groups did not have above chance accuracy. So like, I don't know which way it goes, but let's say training in 9-month-olds and then testing in 12-month-olds, even though that particular point in time, the 12-month-olds were not above chance. We see patterns like that because of the noise signatures. This is my hypothesis, because of the noise signatures. So seeing a lot of noise at train time is great because your model can figure out which features are good or bad, but seeing a lot of noise at test time is bad. And so using data with less noise at test time is a better idea. And so if you train in the noisy condition and test in the less noisy condition, that's my hypothesis about why that might work better than training and testing within one of the two conditions.
Okay, interesting. Thank you.
Sure.
I have a question on the application of the infant speech decoding. And so as you know, EEG has gotten like quite cheap and portable and they're kind of all over the place. And I wonder if you could make an early diagnostic system for speech impediments in children by looking at like the 9-month-old, and if they don't have a latent representation that maps onto what you'd expect, then you might be able to predict early on that they're going to have speech trouble and intervene before it gets bad. Whereas currently you'd have to wait till they're about a year and they just don't start talking. And you might miss some chance to intervene.
Yeah, that's a really great idea and something I'm interested in studying in a little bit older children. So one thing we're exploring is that there are groups of children that they call poor comprehenders. They're very good at reading single words, but they are not great at putting the words together to make sentence meaning. There's just something that breaks down in that system that goes from words to sentences. They read the words, they understand what the words mean, but it's something beyond that. And so I think we could compare brain imaging data between poor comprehenders and normal comprehenders and ask, where is that breakdown? Where do the brain systems start to diverge? And could that help us to tune our therapeutics in order to try to help kids work around that? But you're right that even in younger children would be interesting too.
Thank you. Very nice talk, as always. It was expected from your lab. I was just wondering, you said something about how brain representations compare to language models and visions. I was wondering if you're aware of this new work that came out of, I think, Meta AI. It was by Amii King, and the title of paper is that brain representation partially converge with the language models, something like that.
Did you say partially converge?
Partially converge. Okay. And so the experiment is such that some adults read sentences, and so there's both vision and language perception involved. And they looked at how is the brain similarity with CNN and language models. And so in both vision, lexical embeddings and compositional embeddings, and interestingly, most similarities and like a high similarity was found for the compositional embeddings and not so much for vision. So I felt you said something that I thought it was like in contradiction, like vision is more brain-like in CNNs than language, so.
Yeah, so what I meant to say was that vision is better understood, and part of that is because animals see, and so it's much easier to do invasive recordings on animals than it is humans. And so that's one of the reasons why we understand the visual system better. And language systems in animals, there are very primitive sorts of communications, but there's no language. And so language in animals, you just can't study it. There's no there's no animal model for language. And so our understanding of language comes only from those experiments we can do on humans. And so it's just less well understood, for example. Yeah.
Interesting, thank you.
Sure. Okay, I think that's it. Do you want to say something, Nathan?
Okay, let's thank Alona one more time for a very interesting talk.